Hey, hi everyone. So again, my name is Ari. I'm a third year MD PhD student, so first year graduate student in Dr. Samantha Bell's lab at the Rutgers, New Jersey Medical School. And I'm very excited to share my work with you all today, delving into the interplay between Mycobacterium tuberculosis and macrophage migration. So tuberculosis, or TB, is caused by the intercellular bacterium Mycobacterium tuberculosis, or MTB for short. And MTB has been a human pathogen for thousands of years, but in popular perception, a lot of people consider it as a quite antiquated disease, inflicting deadly illness throughout human history and causing the greatest number of TB-related deaths in the 18th and 19th centuries. But it wasn't until late into the Second World War when Selman Waxman and his then graduate student, Albert Schatz, discovered streptomycin on this very campus, an antibiotic that greatly aided in the fight against TB in the 1940s. However, despite the accelerated growth of antibiotic discovery in the mid to late 1900s, the recent rise in multi-drug resistance have made treatment of TB today difficult and inefficient. And while we don't commonly see TB in our daily lives in the US, it remains a serious global issue as it infects about 10 million people and kills 1.5 million people annually. And in fact, recent increases in TB cases here in the United States has all the more emphasized the need for better anti-TB treatments and therapies. And these reasons have led to recent research to focus on the identification of novel targets for the development of host-directed therapies that augment our immune systems to better fight infectious pathogens. And this requires a robust understanding of the host-pathogen interface. MTB is an example of such pathogen that has evolved clever ways to subvert the host cell antibacterial defenses. And when MTB is transmitted during infection through aerosolized droplets, it travels to the lungs where they get phagocytosed by surveilling macrophages. And while macrophages are traditionally well equipped to destroy infectious and invading pathogens, MTB is able to poke holes in the phagosome to secrete more than 100 effector proteins that are implicated in disrupting host antibacterial pathways. And one host cell function commonly targeted by intracellular bacteria is the actin network. And the actin network is responsible for several important cellular processes, which I'm sure all of you are aware of, such as maintaining cell shape, facilitating vesicular transport, forming protrusions for phagocytosis, endocytosis, and cell migration. And if we focus specifically on macrophages, its ability to migrate is essential for many of its functions. So macrophages use actin-based motility to migrate to different tissues and organs during infection, surveil for nearby pathogens, and move to sites of antigen presentation to adaptive immune cells. And the actin network is a dynamic structure that is made possible by the regulation of actin polymerization, which turns globular actin monomers to filamentous actin polymers. And this involves small GTPases like RAC1 and CDC42 to activate actin nucleation promoting factors like WASP and WASP and WAVE that then activate the ARP2/3 complex, which directly generates the newly branched actin. And these regulatory proteins have been reported in literature to be targeted by intracellular bacteria and through their effectors, which led us to reason that such targeting of actin regulators can happen with MTB in macrophages, especially since the molecular mechanisms of how MTB infection alters macrophage actin network remain incompletely understood. So we ask, does MTB change macrophage movement during infection? And do bacterial effectors target actin, the macrophage actin network? Therefore, to interrogate how MTB alter macrophage migration, I hypothesized that MTB infection inhibits macrophage migration by targeting actin regulators, thereby affecting downstream functions like bacterial surveillance and moving to sites of antigen presentation to promote infection. So to test this hypothesis and first study gross migration, I developed a scratch assay adapted for murine macrophages that we use in our lab, such as the immortalized bone marrow-derived macrophages, or IBMDMs for short. And a scratch assay is essentially a technique that measures cell migration by tracking area repopulation of a scratch by a monolayer of cells over time. And I start by growing cells in a 24-well plate to confluency and serum starve these cells for 24 hours to prevent cell proliferation. I then make a scratch on the cell monolayer with a sterile pipette tip and leave these cells in serum-free media to be imaged over time. And preliminary data of baseline macrophage migration shows cells efficiently repopulate the scratch over the course of 3 days. And I generated the scratch leading edge lines using a previously published wound assay plugin for ImageJ, which analyzes these scratches in an automated and unbiased fashion. However, these are unactivated IBMDMs, and activated macrophages have been reported to exhibit a stronger migratory phenotype than unactivated macrophages. So to test this, I treated cells with a TLR2 agonist PAM3CSK4 to activate my macrophages, and I found that PAM3CSK4 treated cells repopulated more of the scratch over time than untreated cells. And when I quantified the areas repopulated, which is seen in the graph on the right, I found that activated cells migrate significantly more than untreated cells by 48 and 72 hours. And it is important to note that infection itself can activate macrophages via TLR2. However, we are using this immune agonist as a means to validate the scratch assay rather than mimic any activated macrophage biology during infection. However, this not only demonstrates that our scratch assay workflow can efficiently measure unactivated and activated macrophage migration, but it also suggests that PAM3CSK4 can serve as a promising control for future scratch assays. And having looked at gross macrophage migration, I then wanted to focus on potential protein interactions between MTB-secreted effectors and macrophage actin regulators that might induce changes in macrophage migration. And one MTB effector candidate that I identified from literature is PPE15. So one paper reported that PPE15 has a region in its C-terminal that share high protein sequence similarity with SH3 domains commonly found in eukaryotic proteins. And the SH3 domain in eukaryotes is a highly conserved protein motif that takes on this barrel shape that comprises of several antiparallel beta strands. And this domain binds to proline-rich regions, and many actin regulators, like the actin nucleation promoting factor NWASP, has proline-rich regions that are known to interact with SH3 domain-containing proteins. So using the AlphaFold3 server to get a sense of PPE15's predicted structure, I noticed that there are two predicted beta strands early on in the SH3-like sequence. Therefore, I hypothesized that PPE15 may interact with NWASP through its SH3-like domain. And to see what an interaction between PP15 and NWASP might look like, I used the AlphaFold3 multimer prediction server. And interestingly, we see PP15's disordered tail seemingly wrapped around NWASP's looped structure. And when I used Chimera X to identify the protein sequences of these regions, I found that PP15's predicted SH3-like domain and NWASP's proline-rich region exist in very close proximity, which we found really interesting. So having used AlphaFold as a predictive tool, I performed a preliminary co-immunoprecipitation assay of flag-tagged PP15 and HA-tagged NWASP using a flag pull-down in HEC293 T cells. And I used anti-flag beads to pull down my flag PP15, and I used flag GFP as my negative control. And as expected, GFP did not interact with NWASP. However, as highlighted in yellow, I did see that PP15 did bind to NWASP in this Western blot. So not only does this demonstrate that structure can be used to predict and model protein interactions, but it also demonstrates that PP15, which is an MTB effector, can bind to macrophage actin regulators like NWASP. So taken together, I'm very excited to proceed with this project, focusing on primarily four next directions. So the first being performing a scratch assay with MTB to determine whether infection alters migration, making PP15 expressing macrophage cell lines and performing a scratch assay with it to see how PP15 might affect actin-based migration, making PP15 mutants lacking its SH3-like domain to determine if this domain is primarily responsible for its binding interaction with NWASP, and performing a scratch assay with an MTB strain that's mutant for PP15 to assess the potential role of PP15 in infection-induced migration changes. So I'd like to acknowledge the incredible people I get to work with every day in the Bell Lab. I'd also like to thank Samuel from the Lutz Lab for guidance with how to analyze scratch assays, and James from the Patrol Lab for teaching me how to use AlphaFold and Chimera X. And with that, thank you, and I'm happy to answer any questions that you might have.
I mean, instead of looking generally at grade 7 knockout and just asking whether, just in general, that you see this kind of migration effect.
Yeah, so I'm not sure, like in literature, if anyone has done that specifically to look at macrophage migration, but that is something that we are considering to look into when we're working with like our wild type MTB strain, and we have our like MTB. We're trying to develop strains that are mutant as well. So looking at a MTB strain that is mutant for its secretion system will also be interesting to look at to see if that has any effect on actin network and macrophage migration. Thank you again. Thank you.